Hereby, I open the academic ceremony in which Till Steinbach will defend the academic thesis Making Tactile Sense, mapping the path from research.
This is entitled Making Tactile Sense, mapping the path from research toward clinical use. In this presentation, I will summarize the work included in my thesis. As the title suggests, it focuses on the sense of touch, how it is represented in the brain, how it changes, and how methods developed in research can be used or can be moved towards clinical use. Touch is essential for everyday actions, such as lighting a match, where tactile feedback guides your movement, especially during fine finger manipulation. In this video, you can see how much the participant struggles when tactile feedback is removed through a local anesthesia of her hands. Even though the muscles are intact, movement becomes slow and imprecise. Clinically, this is important because what may look like motor impairment is actually driven by sensory loss. In many clinical conditions, motor impairments and sensory impairments go hand in hand, and together they substantially worsen clinical outcomes, as seen, for example, after stroke. In the brain, touch information is processed in an area called the primary somatosensory cortex, or S1, which lies directly next to M1, the area responsible for motor output. Within S1, the entire body is represented in an organized map, with neighboring body parts represented next to each other. Importantly, this map is not stable and not static. It changes with use and injury, making it a very promising target for understanding rehabilitation. Within S1, the area that is studied most extensively is the representation of the fingers. And because these also have clinical relevance, because regaining ability to move your fingers significantly improves quality of life, my work also focuses on the finger map of S1. Having access to this map at individual patient level could be clinically relevant in several ways. First of all, it could help identify and track recovery over time. Secondly, parameters extracted from the map could help guide intervention, for example, identifying where in the brain a specific intervention should be applied. And at the same time, these maps could support tailored therapy by adapting rehabilitation strategies for individual patients' somatosensory profile. The key question, or one of the key questions guiding this work, was how we can access these maps in individual patients, which brings us to functional MRI, which gives us the possibility not only to create images of the living human brain non-invasively, but also see where and when it becomes active. When we combine this with the stimulus that the participant or patient can feel on their fingers, we can identify the finger representation in the individual brain. This approach works very well in research, but the key question is whether it can be adapted for clinical use. For clinical use, mapping needs to meet several requirements. First of all, it needs to be fast and efficient enough so it can be integrated into standard MRI assessments without additional burden for the patients. We have to take in mind here that any additional measurement means for the patient that they have to lie in the scanner for longer, which may be uncomfortable for some, or even painful, or just annoying. Moreover, in clinical settings, anything that requires extensive manual analysis will be difficult to implement. Therefore, at least parts of the analysis should be automated. To be clinically useful, outcomes need to be reliable across measurement sessions. Otherwise, it becomes impossible to say whether the observed change is actually due to, or is actually a true cortical change, or mere due to measurement variability. And finally, the mapping needs to be sensitive enough to detect small but meaningful changes, because clinically relevant changes may be more subtle than dramatic. We therefore took established finger mapping protocols from research and adapted them to meet these constraints. The first adaptation was the use of tactile stimulation without active movement. So instead of asking the participants to move their fingers, we attached these modules to their fingers and they could feel the disc vibrate. In addition, we reduced the total scanning time to under 15 minutes by using a paradigm in which all fingers are stimulated within the same measurement run. Using a partly automated analysis pipeline, we were able to extract significant activity in S1 in all our healthy participants. This shows that the adapted mapping procedure is capable of producing meaningful activity that can be extracted in a at least partly automated way. We next wanted to investigate how stable these activations are, and therefore we applied the same measurement or the same procedure in a second measurement session and compared the results. What we found is that the volume or size of the activation varied substantially across sessions. Thus, we concluded that activation volume is not reliable at the individual participant level. This was not entirely surprising to us because activation volume is influenced not only by the underlying neural activity, but also by factors such as attention, physiological state, or even head motion during the measurement. We then examined the location centers of the finger representation, and we actually found them to be quite stable across measurement sessions, despite the underlying variation in activation volume. We concluded thus that the location activation is a highly reliable measure. We next asked whether the mapping depends on the scanner itself, or more specifically on the magnetic field strength, which is measured in Tesla. In general, scanners with more Tesla provide more signal and therefore allow for finer spatial detail. However, they are more expensive and also more challenging to use in a clinical setting. We found again that the location of activation maps was stable when comparing measurements at 3 and 7 Tesla. In contrast, activation volume remained variable. From a clinical perspective, this indicates that, of course, depending on your specific question, lower field strength scanners can provide adequate results already. In the next step, we applied the mapping design to 2-stroke patients. This is an important step for demonstrating the clinical feasibility of the mapping procedure. And we actually also found in the first patient that we were able to obtain a full digit map that also looked comparable to those observed in healthy participants. However, in the second patient, we were not able to detect any significant activation using standard statistical thresholds. We have several possible explanations for this, which range from differences in the severity of clinical symptoms to differences in scanner conditions. Importantly, this does highlight that translating even robust research methods into the clinic is not without challenges. The next test was whether the approach is sensitive enough to detect small changes within the somatosensory map. To do this, we used short-term immobilization of the arm as a controlled model of reduced sensory input, which is clinically relevant because also in stroke, you of course have less sensory input. We applied our adapted mapping protocol before the immobilization and after 48 hours of immobilization. And then we looked at the volume or size of the resulting finger activation maps. And we found for the non-immobilized hand, which is shown here in cyan, we observed a decrease in activation volume following the immobilization. In contrast, the representation of the immobilized hand, shown here in orange, increased after immobilization. Now, some of you may be thinking, didn't he just explain that activation volume is not the most reliable measure, and this effect might therefore not reflect immobilization at all, but could be due to measurement variability? And yes, this is a very fair point, and also why we added a third measurement session to be able to distinguish between effects from the immobilization and effects from repeated measures. And what we found is that actually, without immobilization, the representation of both hands, the volume of the representation of both hands decreased over time, which actually fits quite well to what we see in the non-immobilized hand, because there we also saw a decrease after immobilization. So we concluded that this effect was likely not due to immobilization. However, it is it doesn't fit with what we see for the immobilized hand, because here we see an increase in volume after the immobilization, which runs against the general trend of decrease in activation. Taken together, this shows that the adapted mapping procedure is sensitive enough to detect subtle, clinically relevant changes at the group level. However, when we examined individual participants, we saw that the immobilization effect was highly variable and became a lot more blurry. For clinical relevance, it would be crucial to detect meaningful changes at an individual level, which remains challenging. In some clinical cases, mapping may not be possible, for example, because body parts are missing. In such cases, it may be still important to know the location of the somatosensory finger area. for example, to deliver tactile information through prosthetic devices or to support anatomical or connectivity-based analyses. Therefore, we explored whether anatomical landmarks could help localize the finger area. Specifically, we focused on the central sulcus, which lies directly adjacent to S1. From this extracted central sulcus, we defined 2 landmarks that could be identified reliably across all our participants. For visualization purposes, we averaged the central sulci of our participants, resulting in this cloud-like shape. And we then projected the center locations of the digit activation onto this averaged central sulcus. And what we found is that our first landmark was frequently associated with the representation of one end of the finger area, while the other landmark was more often associated with the other end of the finger area. So together, these landmarks roughly indicate the medial and lateral boundaries of the finger area in S1, showing that even when functional mapping is not possible, anatomical landmarks might still provide a spatial reference. So to conclude, this work shows that somatosensory mapping can be made clinically feasible. By being fast, we managed to obtain meaningful results at the individual level with scanning times under 15 minutes, supported by an automated analysis pipeline, reducing the workload for clinicians. Reliable, depending on the parameter of interest. Location, as we have seen, was very stable, whereas volume was less stable. And it is sensitive to small changes at the group level, which is already very important for clinical research studies. At present, however, this sensitivity could not be demonstrated consistently at the individual level. And to conclude in one sentence, touch can be mapped reliably and meaningfully, and this has clear potential relevance for the clinic. Thank you. And with this, I give the word back to the prorector.
Thank you very much for this very clear and on-time presentation. The opposition, we will start now with the opposition, which will be opened by Professor Bernadette Jasma, was Professor of Cognitive Neuroscience of Language Processing at the Department of Cognitive Neuroscience at the Maastricht University. Professor Jasma is also chair of the assessment committee.
Thanks, Prorector. Dear candidate, first of all, let me congratulate you and your supervisory team to this beautiful work. I read it with great interest. And I think you put it nicely into words in your summary in your thesis. You revealed sensory motor maps, you read the maps, you navigate the maps, you supplement the maps, and you transferred it into clinics. I think you can be very, very proud. And given that I remember some of the lunch discussions at the 3rd level in our building, I knew that you sometimes had some doubts about the sense of everything, but I think your work makes a lot of sense. It's already published, and I think you're placed well now somewhere else, but I think you take this knowledge with you, and you can be proud. But of course, I also have questions. And I frame my questions around the chapter 5. It's a functional MRI on the healthy volunteers where you actually immobilize the hand and the arm. I like the study very, very much. And of course, there you showed that you find this increased volume of both in contralateral BA3B, right? And you provide also nice interpretation of that. You also say we cannot be fully sure, but eventually it relates to excitation of the sensory motor pathway, especially the N9 proposed by Ikeda. It could also be related to elevation of attention or some sort of rebound after sensory deprivation. That all makes sense. But I would like to discuss with you a 4th thing, because we are motor, you are research master alumni, we had a sensory motor cortex course, and I thought, he didn't mention the feed-forward model at all. So I would like to see whether there's an alternative explanation, and it relates to the task design. So in the scanner, you simulate sort of sequentially from one finger to the other. And if I would be the volunteer, I can predict where the stimulation comes next. And then there comes the gap. And would that be something like a violation of a prediction? And that would lead to elevation of error signals, and that could explain the elevation in the bolt that you found. And then, alternatively, when I then look into the control condition, the other hand, where you find a decrease, makes also sense according to a forward model, because you would expect sensory-induced suppression when everything is according to expectancies. I would like to hear your thoughts on that.
Highly esteemed opponent, thank you very much for your compliments and your question. I think this is a quite interesting idea. I'm not sure if I fully understand the theory you're proposing, because I think what you have to keep in mind is that the immobilization was never the first session in this paradigm. So at least once the participants already perceived the stimulation. And the stimulation didn't change between the sessions. So I never talked to the participants about it, but I can imagine that after the first or the second, because it's also same across their hands, runs, they already know what to expect. So they know there are stimulation, they move from one finger to the other in a sequential order, and then after 4 stimulations, there's a break, and then it continues again.
Yeah.
So of course, there could be some sort of prediction going on. This should be the same across hands because it's the same, right? So also for the non-immobilized hand, we have this prediction going on. And it should also be the same in every session. Of course, you can argue in the first session they didn't know that, so there's less prediction. But then this would mean, especially for the immobilized hand, that there would be less activation coming, or you would see less volume of activation.
So I'm with you and you're fully well on track. But when I'm immobilized, yeah, there's something weird. So, yes. And how you discuss in the chapter is basically like more like a bottom-up process. So you stimulate and then you measure what happens. And my question relates to, well, is there more going on in area 3B, 3B, where actually you not only measure feed-forward processes, but also eventually top-down and predictive coding? Could that be possible?
Yes. I totally agree that this is possible. And this is also what I meant when I was writing that, of course, after immobilization, everything that happens to the previously immobilized arm is new kind of to you. I also actually experienced this, and I noticed in the scanner that the stimulation felt a bit different, not necessarily painful, but more intense. And I think this is just because after 2 days, rarely feeling anything to your arm, it becomes more, let's say, interesting that something is happening to your arm, and therefore you automatically put more attention, which of course is higher level input into S1, and therefore we see more signal. That's currently my working hypothesis.
Yeah. Yeah, and one future thing would be then how to disentangle the different processes at one point, and to see when in patients whether actually you have more deficit in bottom-up or also in the error monitoring. Do I have time for a little one? So related to that, you also did so nicely the psychophysics or the control experiments in behavior. And there, you did the finger lifting. So there is a color code on one of the fingers that are displayed, and that is the instruction to the people to actually lift one of the fingers. And there you do not find differences between immobilized and normal hand. And I was wondering why that is, and I would like to ask you about your point. Might that be related to having 2 different types of tasks? The one in the scanner also allows prediction, whereas the other one does not at all.
Yes, this could be, but because based on previous literature, we were surprised not to find any results, because immobilization should affect reaction times, regardless of whether you can predict what is happening or not. I think the problem here is more that the behavioral tasks were performed after the MRI session, which took 1.5 hours and involved stimulation to the fingers, so maybe already redoing all the immobilization effects. It would be interesting to see, without the MRI session, whether we see actually behavioral results. And we did, during COVID, actually, an online pilot study on immobilization effect on reaction time, and there we did see an effect. So I highly assume that the not seeing an effect in this study is because of the time between the end of the immobilization and the start of the behavioral measurement.
Okay, yeah, thanks a lot, clear answer. Give the word back to the prorector.
The opposition will be now continued by Professor Liane Kari. She's Professor of Rehabilitation and Clinical Neuroscience at the School of Allied Health, Human Services and Sport and at the Maastricht University in Melbourne, Australia. She's online.
Thank you very much. I'd also like to pass on my congratulations for this sustained and original work that helps to connect the science of sensory processing with its potential application in stroke clinical practice. And from this perspective, I really enjoyed it as both a neuroscientist and from a clinical perspective, in which I do work with a number of stroke survivors, particularly those who experience sensory loss and have done for the last 30 or 40 years. I'd also like to acknowledge your main contributions, which is the development of the fMRI digit mapping protocol for clinical use, looking at the somatosensory plasticity with the neurotypical participants, and reaching further to aligning the structural with the functional landmarks, particularly given that it may be an avenue as a proxy if the functional imaging doesn't work. I'd also like to just highlight your two key strengths that I got from your work. One is the high academic quality in the writing and methodology, and the fact that you did look at new insights rigorously with supported evidence. And secondly, in your creativity in the approach you took, which really came through in relation to the protocol development, the themed summary, and also just more recently, your making tactile sense image that you put on the front page of your revised thesis. And I think there'll be a lot of stroke survivors who would really connect with that. So thank you for sharing your journey in that way. The question I'd like to focus in on is that one around developing viable functional MRI protocols for clinical application, recognising it's challenging, especially with stroke survivors with varying impairments. One of the advantages that you clearly of your protocol was that it was developed not to require active movement during tactile sensing. I would, however, caution that being able to capture the active process of tactile information processing, that it actually needs to be attended to. So you refer to your task as passive tactile stimulation. I just want to caution you that in clinical practice, this would typically be interpreted as a passive task also for the client. So considering this, I'd really like you to critically reflect on the specific features of your developed protocol and how they do or don't map to active, attentive sensing of tactile information.
Highly esteemed opponent, thank you very much for your compliments and also your question. Maybe just a quick question, because I'm not sure if I understand the question in the end correctly. It is about the adapted finger mapping design and the use of what I call passive tactile stimulation. Whether this, or what you're saying is that basically, from a clinical perspective, both are highly relevant, to have the passive part, but also to see what active movement actually does in the primary somatosensory cortex?
No, that's not quite what I'm saying. I'm saying that attentive processing is an important part of sensory processing, whether it's active or passive. And when a task is referred to as a passive task, whether or not the task also requires active attention or not is really quite critical, both in how it's presented and how it's interpreted. And so it might come down to specific small features of how you introduce the task, the type of instruction, or picking up whether the person did or didn't actively attend during that imposed tactile stimulation or the passive stimulation. So it's getting to that very fine level of looking at the features of the fMRI task and controlling for attention versus passive, etc. So I really want you to critically think about what your task is or isn't achieving and mapping in terms of the information processing.
Yes, I fully agree with you that because we're using fMRI, right? So what we are measuring is input into the primary somatosensory area. And this input could either be bottom up, so basically from the tactile sensors of your fingers, or it could be top down from attentional processes happening in the brain. And me calling the stimulation method passive tactile stimulation doesn't fully capture what it is. I agree with this because we asked our participants to attend to the stimuli and we actually built an attentional task, so they had to count little interruptions in the stimulation. The reason we called it passive is to differentiate between what we and what is broadly known as active mapping, which involves the participants to move their fingers. So we said passive as to involve there is no active movement necessary. Of course, this means that in our healthy participants, there is still attentional demands. And in a clinical setting, I know that this cannot always be assumed that is there. Sometimes attentional networks are destroyed or don't work as well, and therefore you cannot ask all patients to pay attention to the stimulus. Maybe they don't even feel it, then you can't even attend to something you cannot feel. I think it's still relevant to have this passive or this attended passive stimulation in healthy participants, because we know from previous literature that attention to the stimulus also increases activation. And we do want to see activation, and we do want to see stable activation over sessions, because if we don't give if we don't say to our participants, please attend to the stimulus and have a task, then we don't know if they attend to it, if they ignore it. And this could also change between sessions, making it more variable in what we see. And I also think from a clinical perspective, if I remember correctly, there is some evidence that if the attention recovers, then also it's more likely that the somatosense will recover. So this could also be something we can see. Okay, I'm seeing you're not completely agreeing with what I remember.
It doesn't, I continue.
Because if we see changes in the somatosensory map, this could either be because of some top-down effects or because bottom-up effects, right? And if we, for example, would know that attention helps in rehabilitation and we see an effect in our maps, then this could be an attentional effect, but this would be still interesting in a clinical setting.
Great, thank you. It's really important, and I appreciate that you are seeing both aspects there. I think if you are looking ahead to communicating it, being able to say something like tactile stimulation without active movement, but with attention is important. I know when we've done touch discrimination activation with fMRI, if we're not very specific in both the instruction and afterwards, like a visual analog scale of how well they attended it or not, even in healthy individuals, you lack that activation. And we have done tactile discrimination in 100 or more stroke survivors where they can actually then report that level of stimulation. And I think even as in your response to the last answer, attention to the task could also be an alternative explanation for some of the things that are going on, and could also be for the sensory where you did see in one and not in another. So I think a really critical part of the whole task. So thank you for your response to that, and I pass back to Prorector. Thank you.
Thank you very much. The opposition will now be continued by Professor Lotze. Professor Lotze is Professor of Clinical Neuroscience and Plasticity at the Universitätsmedizin Greifswald in Germany, and he is online.
Yes, thank you for this very nice work, and I really appreciate the whole comprehensive work, and especially the tasks which were done in healthy participants, especially the immobilization task, which really should be published. I had some problems with the power analysis of the pilots, I would say, with these high field strengths comparison with just 3 participants, and especially with the investigation on stroke patients. And my question as a neurologist would be, why did you select these capsula interna or subcortical lesion patients for the task?
Highly esteemed opponent, thank you very much for your compliments and for your question. I mean, the honest answer to this is we didn't select them, or I didn't select them. These were selected when I started. And from what I gathered, or from also what I know, is in clinical research, you cannot be too selective about your patients, because otherwise you cannot finish your project in the time you want to finish it. From what I understand, these patients, they followed a specific rehabilitation strategy at the center we were collaborating with, and they agreed to participate in the study. That the underlying lesion was in similar places, I think, was a mere coincidence. And our idea behind this study was also more, or the idea behind the study was more that we wanted to test whether our approach works in a broad clinical sample. So the idea would have been to not select patients based on any lesion area. The idea even was that we wanted to include severely or patients with severe somatosensory deficits, to be able to show that even on cases where with standard assessment of somatosensory you would have problems because they don't understand. I mean, we didn't put non-understand because we also need them to understand to sign the informed consent form, so this might be a bad example actually. But we wanted to have severe deficits because what we wanted to show is that our design can still work even in severe cases and extract useful information. I'm not sure if that answers your question.
Yes, it does. I know the problem there. Yeah, that's usual. Thank you a lot.
You can ever, do you have other questions, or you have a bit more time if you want, otherwise we move on.
Yes, if you do larger stroke studies, you know that problem, and it's really a pilot here, so I'm quite confident with the answer. But it's really a huge challenge if you transfer this paradigm to stroke patients. It's much easier with chronic pain patients. We use this paradigm, but with stroke patients, it's really challenging.
Maybe a note on that, because I do agree that we have to be very careful here in how we interpret the results, because having maps from two stroke patients, there is only limited interpretations we can take from it, because two patients cannot speak for the whole population of stroke patients, which is quite the heterogeneous patient population anyway. So I agree we have to be very careful with what we say based on the results we have, because as you said, these are just pilot studies, and there needs to be like a larger cohort study where also things like you said, lesion location and different aspects of deficits have to be taken into account in order to actually say, yes, we can use our mapping designs in stroke patients.
Thank you.
Thank you very much. We will continue the opposition now with Dr. Anne Mulders. She's expert in clinical neuroscience and plasticity at the Experimental Health Psychology Institute at the Maastricht University.
Dear promovendus, first of all, of course, I also want to thank you and your supervisory team to invite me to be in. To invite me to be in your doctoral defense. I have read your thesis with a lot of interest. It's well written, and I also particularly appreciated the witty reference to Harry Potter's Marauders map, and that comes through the entire thesis, so that was quite entertaining. I see that you worked hard. You developed sophisticated methods to investigate a timely and clinically important topic, and your efforts also have been rewarded. One study is already published, and also you are here to defend your thesis, so well done. I already want to congratulate you and your supervisors on this achievement, but of course I'm not only here to give praise, and I also have some critical questions. So first of all, I might also want to continue discussing a little bit chapter 5. I thought it was a cool study where you try to have this short-term immobilization to see whether it would affect some of the sensory plasticity of S1. Now, you found some effects on volume. You already mentioned that was smart in your presentation, that you have to be a little bit careful because the retest reliability of that particular measure was not the best one, let's say. So I was also wondering, why did you decide to go for 48 hours of immobilization? Because you mentioned in your introduction that there were previous studies going up to 23 days of immobilization. Okay, you also mentioned that one study was successful in having only 24 hours immobilization. But I just wondered, why did you choose this time frame? And do you think that, given that you also mentioned already, replying to one of the other opponents, that you think that when you actually tested for the behavioral tasks, that the immobilization effects might have already disappeared? So would a longer time frame have also led to more long-lasting effects or not? So can you reflect a little bit on this?
Highly esteemed opponent, thank you very much for your compliments and for your question. Yes, this is quite the interesting topic because I also, to say this up front, but I will say more about this, I don't know. Mm-hmm. The idea why we went for 48 hours is because we wanted to do short-term immobilization. The paper you referenced with 23 days is actually on, it's difficult to say, but it's not really patients. It's people who broke their arm and therefore had to wear a cast, which of course makes it easier to immobilize the arm for a long amount, like ethically, for a long amount of time. But of course you have also different aspects to consider because you could have still pain in your broken arm, or the cast could also be that you still feel some sensory input. So it's not as controlled as what we could do with our approach. And we wanted to deliberately investigate small changes, which in hindsight might be a bit too ambitious, because now what we have is we have something where we were not exactly sure what to expect, and we have a new design which we want to test. So then, of course, you have the problem that if you find something, or let's phrase it differently, if you find nothing, you don't know. Does it mean immobilization didn't work? Does this mean the design doesn't work?
Yeah, so that's an interesting question. Indeed, is it because in these longer, you already mentioned that a confounder there is that they also had an injury, so is it just immobilization? Would it be enough to result? So longer immobilization, would it have similar effects, or is it because of something additional, like a real injury or something else happening that mixed up some of the sensory function and mapping? So yeah, and then of course it becomes even more interesting when you also show. So now let's assume that your manipulation worked and that it was only relieved because there was quick cortical plasticity and the effects were gone after your fMRI, but that they were still there during the fMRI. You picked up some changes, which you say they are maybe not super reliable, but then it also didn't correlate with any of your behavioral tasks, right? And then the question is like, Why? Is it because your manipulation was not strong enough and actually these small things that you saw in the brain did not correlate and that's a true absence of relationship? Or is it possible that the tasks were not the best task? Also here, my opponent also talked about that. So can you elaborate a little bit what you think is missing there in that part of the study?
Yeah, so first of all, as I said before, I really believe that we should have seen behavioral effects after the immobilization, because the pilot study I referred to beforehand, the immobilization duration actually was, correct me if I'm wrong, Zephyr, I think it was 9 hours. So, and we did see behavioral effects afterwards. So I think, or I believe that after 48 hours, we should have seen some behavioral effects. Therefore, I really think the problem here is not counterbalancing the task, which we did for a reason, because the main interest of the study was the fMRI. So we wanted to have the fMRI directly after relieving the immobilization. So there we would still see the effects. It could, however, also be that the changes we saw, because we did only see them at group, where we also saw in some participants, of course, but these were mainly at the group level. We couldn't see like a specific pattern that we could observe in every participant.
Yeah,
so
but you mentioned, so you already mentioned that you saw these effects in the pilot on the task with the finger lifting, but did you also, so the speed basically, but do you also have similar effects on your tactile acuity, or was that absent in that task, or you didn't have it in the pilot?
The task in the pilot was a bit different because it happened during the lockdown, so we had to do it online, and we had like a keyboard task where they had to press like a sequence of buttons, and we measured the reaction time and the accuracy. So I don't know about the tactile task. I would still have expected to see something there, because also the literature shows us that immobilization actually decreases tactile acuity in the immobilized hand and increases tactile acuity in the non-immobilized hand. But yeah, we didn't see that. I think this has also to do with the fact that the tactile acuity task that we choose it from a different study made by one of my supervisors. And it's quite the lengthy task. So I think combined investigating both hands took around 40 minutes, which of course is quite tiring for the participants after 1.5 hours of scanning. So yeah, I think having like a shorter task, maybe just focusing on 2-point discrimination would have been the better choice.
Which is of course also very unreliable. But do I still have time for one small question? Oh, small, I don't think small, but so I was wondering if, because you actually just already mentioned it, it's a nice way into my next question. You say like your stroke patient that you tested, they showed differences in tactile discrimination after some rehabilitation training. But again, there, I am of course very pro looking into fundamental mechanisms because I'm also an experimental psychologist and interested in basic mechanisms. But what is now the true or let's say I'm not really that convinced about how useful it is in clinical practice and whether it would indeed predict recovery. And you also show in your study that patients that had rehabilitation, they showed changes in the brain, but they did not relate again to functional recovery. So my question is, why is that? And secondly, and more importantly, how do you think that what kind of interventions do you think about if you would want to retrain the somatosensory cortex, or if you want to regain that somatosensory function? What kind of interventions are you thinking about?
So to answer your first question, if I remember correctly, was why I don't think we saw changes related to rehabilitation.
Yeah. So again, is it not there, the relationship, or is it just, would it be a time lag and didn't you measure it at the right time, for example, or just brief thoughts on it?
I mean, there are countless of possibilities, right? It could be that the cortical effects were so small that we didn't pick them up with our method. I mean, we did see changes in cortical activation for the patient we could actually extract data from. Mm-hmm. However, this didn't really correlate to rehabilitation success.
Yeah. The reason I ask is because in your presentation you said that it led to clinically relevant changes, so that's why I jumped on it, of course.
clinically relevant. When did I say that?
Yeah, well, you did. I just noted it down.
Yeah, I mean, what you have to keep in mind, and maybe this answers also your second question, what these designs are meant for is not to be like, I'm finished my PhD now, so we can use them directly for predicting whether stroke patients will be able to feel again or to move again. It's more like a journey, right? And what I think my thesis, or our work shows, is that these methods can be stable and they can be used on patients. And with that, we can do studies on patients. And with that, we can then identify how the map changes and whether these changes or whether any specific parameters from the map can be useful to predict long-term rehabilitation success. Or maybe it's just that we see that, okay, the map are there, but they don't change. But what we can do, we can actually localize where the hand area is, and then we can do some anatomical or functional connectivity studies with it, which then helps us to see whether there is still the potential to recover. So I would say that from my work or from our work, we cannot say whether this is clinically relevant per se, but it could be used to investigate what biomarkers we can use to later predict outcome.
I totally agree with you. Thank you very much. I think our last question we will have to discuss later during the reception, and I will give the word back to the pro-rector.
Okay. The position will now be continued by Dr. Joel Reitler, who's expert on somatic science and motor science at the Department of Cognitive Neuroscience in the Maastricht University.
Thank you. Dear candidate, first of all, my congratulations on your great piece of work. I also really enjoyed reading it. There were actually two things that really stood out to me. So on the one hand, it was already mentioned that you really ventured into multiple domains, right? So you looked at differences across field strengths, plasticity, clinical relevance, and so on. So I think that's great. But the thing that really was impressive, I think, was that for each chapter, you really included a very considerate and frank consideration and description of all the limitations. And I think that should be highly applauded. While reading those considerations, I also came up with a couple of questions. And indeed, my main focus was on chapter 5. So this has been touched upon already a couple of times. But there were a couple of things that I would like to go over with you about this study. So first of all, in the beginning of the description, you briefly mentioned that you also looked at the handedness scores of your participants. And what stood out there was that the range was actually quite wide. So it went from, I think, a score of 5 up until 100. So the dominance for the different participants of the handedness is quite variable. So I was mainly wondering, okay, how could this have influenced your results? Because in the end, in the analyses, I don't think you actually incorporated that information, even though it was available. So could you maybe discuss this a little bit?
Esteemed opponent, thank you very much for your compliments and for your question. Yes, so you're right. Handedness was an inclusion criteria for the study because we only wanted to include right-handed participants. I mean, you probably know, but maybe for the audience, there's like the Edinburgh Handedness Scale, which goes from -100 to 100, with 100 meaning right-handed, -100 meaning left-handed, and 0 meaning bidextrous. And our inclusion criteria was that the score should be above 0, which of course has a wide range of different right-handedness-y. So this could definitely have influenced the results in a way, because what we did is we restricted the dominant hand to have the highest effect. And of course, if people are more used to use their left hand, then the effect might be smaller because they already use their left hand more in an everyday setting. We didn't, as you said, we didn't scientifically test this. However, what I can tell you from the data I have is that all our participants, because we measured the amount they used their arms once during immobilization and then also during like a random time interval. And all our participants used their right hand more during the random interval where they could use both hands except for one, but this was not a participant where a low score was measured.
Mm-hmm. Okay, yeah, that makes total sense. But of course, that doesn't necessarily mean that in the brain it also looks as divided, let's say. So actually, going on with that point, when you look at your results, you have a couple of ideas of why you find this increase in volume for the immobilized hand, right? So you mentioned maybe there's like increased attention because now suddenly that hand becomes available again to the system, or it could be some bottom-up increase of excitability since you haven't used that hand for quite a while and suddenly everything is again online. But I actually thought, okay, maybe there's something else going on. So could it also be that you actually see a form of disinhibition after the immobilization? So the point would be that there are some indications, at least as far as I remember in the literature, is that if you immobilize one of the limbs, then the representation in the cortex gets isolated from the rest of the network. So that could basically mean it could be a reflection that actually the other hemisphere is inhibiting that representation, which is currently not in use, let's say. So could it also be that once you stop your immobilization, that this inhibition that is going on between the hemispheres is now resolved? So you see a disinhibition of the hemisphere that was affected, and therefore also this increase in volume. What do you think about this?
This is an interesting theory. I never actually thought about it in this way, because what I know, or what I think I remember from literature, is that it has been shown that because when you don't move your hand, also the area becomes less often activated, and therefore the inhibition of the contralateral side, or the ipsilateral, from whatever perspective you want to see it, takes over. So basically, as an explanation for why you would see reduced activation is because it's inhibitory, or it's inhibited from the other side because there's more activation going on, because you use the non-immobilized arm more. But for me, this also is made even more difficult because at least I don't fully understand how you would see inhibition in MRI, because inhibition is still input, and therefore I would, like, if the area is more inhibited, I would not necessarily expect there to be less activation, right? Because you still would see inhibitory input coming in. Mm-hmm. Not sure if that answers your question.
Yes, yeah, that's a very fair point. Okay, and then maybe if I still have time, I think so, right? Then I would still add to one more point, and that is the discrepancy between chapter 4 and chapter 5. So in chapter 5, as we already discussed, your main finding is that after the immobilization, you see an increase in the volume of the fMRI activity for the representation in S1 that is coding for the previously immobilized hand, right? But then in chapter 4, you mentioned that for one of the patients, your mapping protocol actually worked out quite nicely. So you were able to nicely pinpoint where the different digits were represented. But you also show that across the 2 sessions that you performed in that patient, that the volume again increases from session 1 to session 2. And then you explain this effect by saying, okay, maybe this is related to the rehabilitation efforts that were done in that patient in the meantime. So that would mean that in chapter 4, you ascribe your increase in volume to a change that is related to neurorehabilitation induced plasticity, whereas in chapter 5, you basically find that with immobilization, you see an increase in your activity volume. So these seem to be contradictory somehow, right? So on the one hand, you have exercise leading to increased volume. In the next chapter, you see immobilization leading to increased volume. So what should we really think about what this increased volume then actually reflects?
Yes, this is quite the interesting question. And I think this can be even, like, I think what would be interesting in this regard is to see how immobilization is affecting the cortex over longer periods of time, because if I remember correctly, the rehabilitation training between the sessions was 3 months, couple of weeks, I don't know, but it was definitely longer than 2 days. So it might be that just the cortical change over time looks different. So for example, if we would now imagine we would have taken a longer immobilization and then measure at certain time points, which of course is not possible because that would break the immobilization, but maybe we see an increase at first and then a decrease, which would also explain why in the study where they had 24 weeks or 23 weeks of immobilization, they did see decrease in activation volume. And the same could be argued about the patient study. So maybe if you have, like if you for weeks do the same training movement, then actually you do see this increase that we see in this patient. I mean, we also see that in the mobilization study, if you do not do any specific intervention in between, we see a decrease of activation after multiple scanning sessions. Why we don't see that in the patient, I also don't know for sure. I think this might be because, now I'm answering a question you didn't ask. But
that's fine. It's interesting anyway, so go ahead.
I think this might be because our participants in the mobilization study, a lot of them, for them it was the first time being in the scanner. And then being in the scanner, of course, everything is new and you have more attention to what is happening. I don't know, but I highly doubt that for the patient this was the first time in an MRI machine. So I think this might also have affected the activation volume of this particular patient.
Yes, very good point. Okay, thanks for the elaboration. Then I think my time is up, right? So I'll give
Thank
it
you
back
very
to
much. We have like a few more minutes. I'll give the word to Professor Jansma.
Yeah, okay, thanks. Enjoyed the discussion. Related to this, and that's a general question, so I remember when I got training some centuries ago in fMRI in Frankfurt, I thought what's going on, a lot of crazy people were running around with prisma glasses because you did the study on what happens when I do reverse left-right imaging, and so it's similar to your immobilization, basically. But I remember that in that study, you looked also into different areas of the brain, like what happens during adaptation to context in primary visual cortex, but also motor cortex. You only looked into S1. And one sentence always is, the brain is a complex system. So why did you choose to only look into one area? And did you have the chance to look into other areas that actually might interact with S1 and whatever you found?
Yes, this is also a very interesting and important question. I also thought about this, also for like thinking about what to answer. I do fully agree that it is always dangerous to look only at like individual areas of the brain without considering that the brain is a network and everything works together. The reason we choose to look only in one area is because we had to start somewhere, right? So because the ultimate goal of our work was to have a design that works and we can extract information. The next step would then be to see how useful this information is. And as I think I alluded before, if I would have to answer this, I'm not sure if the mapping procedure as it is useful or is, I mean, useful, yes, but it's not sure that with this we can predict anything clinically. But it definitely can help us and see what we can use for predicting somatosensory recovery. And for this, I think it's really important to look at the brain as a network. That's why I said maybe it's good to then, from the finger area, to look how it is connected to other areas in the brain. And I think there are really nice studies showing that if you look in patients in the connectivity of the brain, you actually see that if the connectivity to other sensory areas, for example, is changing or is improving, then also recovery is improving in these patients. So I think, yes, definitely look at networks.
Yeah. Thanks.
You may conclude the sentence.
I'm done.
Thanks. So, till Steinbach, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberation and our return in this room.
All right, please.
Till Steinbach. The Greek committee, the Greek committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualification, the degree committee has decided to grant you the degree of doctor. Professor Gebel is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Till Steinbach, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Before I hand over to Renate Schweitzer for the laudatio, I just want to say personally how proud and happy I am that you finished so successfully this journey and now are Dr. Steinbach, and I'm happy to say this first. You will hear it a lot. I also want to say thank you to the Corona. Thanks all for coming and for having such challenging questions for Till. And I also want to especially thank our remote two opponents, Professor Carey and Professor Lotze. Thank you very much for joining today. And I also want to say to the family, Sie können sehr stolz sein auf Ihren Till. Es war eine super Leistung heute. But now I want to give credit to the amazing work of Renate Schweitzer in the supervision, and you have the honor to give the laudatio.
Congratulations, Dr. Till Steinbach. An impressive thesis and performance, as everybody agreed on also in the back room. This is an important day for you, Till, and a good opportunity to take a look back. And before I start, I have to emphasize this laudatio should have held by Amanda Kaas, your primary supervisor, and especially today, deeply missed. It began when Till, being a research master student here at the Maastricht University, took one of Amanda's courses. And he was so impressed by her knowledge and by her way of approaching science that he decided to do a master thesis on somatosensory mapping under her supervision. Me, being a long-standing scientific collaborator of Amanda from Göttingen, still remember her excitement during one of my visits in Maastricht of having a master student being interested in somatosensory mapping. Rainer also recalls Amanda telling him about the smart student that would be not only a promising PhD candidate, but also appropriate for a research talent grant. That Amanda recognized Till's specific talent right away is expressed in the sentence that she pushed him to add to his grant application. Quote, "According to my supervisor, I possess the rare ability to ask the right questions." End of quote. This competence is complemented by his personality. Till is often described as a calm, rational, and modest person with a high interest in knowing and a strong drive to understand, which makes him a wonderful person to work with. After the completion of his master thesis and while the grant application was still pending, Till had the opportunity to spend 9 months at the Maastricht Institute in Göttingen, working on methodological details of tactile stimulation under my supervision. I thoroughly enjoyed the deep scientific discussions about chances and limitations of the different analysis approaches with Till and Anna, the PhD student he has now collaborating with. I also learned that Till is a regular long-distance morning runner, and he loves bike rides, as he adapted his work schedule accordingly. This was also the time when he had to prepare for the second round of grant interviews, challenging him and his modesty. In an email to Amanda, he wrote, "The quote, the questions are slightly different than last time, more focused on the topic and not on the person, which I like." End of quote. He got the grant, and together they started with the fMRI-based mapping and the immobilization approach, while Amanda kept building up the collaborations with the Dutch rehabilitation clinics. Then came Corona, and Amanda and Till impressively adapted in keeping the scientific projects running, even together with students in a research practical course, and being awarded with a prize for, quote, designing the most creative experiment considering the limitations of a lockdown during a pandemic, end of quote. But then came the most unexpected, almost unbearable turning point. Amanda passed away 4 months after being diagnosed with an intense and incurable illness. Nothing was as before. Till could have quit, but he did not. Being already deep into the PhD project and having overcome several major obstacles, Till faced the enormous challenge of this situation. Amanda had asked me if I could take over the supervision of Till's PhD project, which I agreed to without any hesitation. But for Till, this meant that the clinical part of his PhD could not be completed. Now, let me come back to Till's thesis and highlight Till's usage of the Marauder's map as a metaphor to illustrate the scientific approach to tactile mapping. I will now use the development of the figure of Harry Potter as a metaphor for the developmental advances Till had to make in the second phase of his PhD. The first step was moving from executing tasks to taking responsibility for action. That meant to be fully responsible for the progress of the PhD projects without a local supervisor being present. His reaction to this challenge was to keep up continuity on different levels. He completed the immobilization project with the help of a motivated bachelor and a dedicated master student that he supervised by encouraging to think further, by giving opportunities to contribute to the design of the study, and by allowing them and him to gain first experience in doing research. But the responsibility extended beyond the experiments. Till and Maite kept up the SOPA lab meetings established by Amanda, the lab feeling, and the dinners. Among the fellow PhD students, Till was known to be always ready to help, to discuss scientific ideas, and to provide useful and honest input. All these components blended together in the second step of the development of scientific judgment. Till's critical appraisals of scientific approaches, measurements, results, and their interpretation is never subjectively biased and always based on careful consideration of the complexity of the topic. This can be seen throughout his entire thesis and makes it a deeply grounded scientific work. Another strength that Till has even further developed in this third step is the handling of methodological and analytical power. He thrives on finding simple solutions to address a question or to build an analysis pipeline. And he's very careful not to overinterpret a result. The last and fourth step is the acceptance of uncertainty. Acknowledging that a research topic or question is worth taking the risk of failure is a hard one. Amanda and Till were ready for that. But it is still challenging when the results of an extended study are difficult to interpret. Till masters this situation by harnessing the methodological power and a good scientific judgment. But all this could not have been achieved without a supporting family and a network of friends. Friends to hang out with, friends to talk to, friends to bike or run with, friends to play board games, friends to live with, and friends that are just there, no matter if in Maastricht, in Düsseldorf, or elsewhere. As a supervisor, I'm very grateful for this essential support, and I hope, no, I'm sure all of you have profited as much from him as he has profited from you. Dear Till, with your rare ability to ask the right questions that Amanda saw from the very start, and the remarkable developments of already laid out traits during your PhD time, you are now at the point where I'm very proud to say that you don't need us anymore. Not because you know everything, but because you know how to proceed when you don't. Congratulations again, Dr. Steinbach.
Thank you very much. I will now close officially the session, but before that, I would like to give you some instructions. Well, most of you are familiar with the things here, but after I will close the session, we will stay here for some photos and also for saying hello and goodbye to the people online. So you can proceed to the rafter, which is on the right, and just wait for Till to arrive there, which will be in a few minutes. Okay. So, dear Dr. Steinbach, also on behalf of Maastricht University and the Faculty of Psychology and Neuroscience, I want to congratulate you with the degree you have acquired. You did a great job. I mean, we are commenting that the translational work is always very hard. Right? I mean, it's abandoning a little bit the easy part of fundamental science and going in the real world. And so very good that you did this. And congratulations very much with your performance today. And hereby, I officially close the academic session.
Hey, hi. I think we are not needed anymore.